the next presentation, and it is microbes and human gut, and it is Anna-Maria Pajari from University of Helsinki, so please.
Hello everyone, nice to see so big an audience. So I'll come from the work package 5. So as we in SIMBA, one of our big aims is that we'll promote the sustainable food system. And just to remind you that around 3 weeks ago, Stockholm Resilience Center gave a new sort of like warning that we have this far exceeded 6 of the planetary boundaries, when compared to 2009 there were only 3 boundaries exceeded. So we really have an urgent need to promote sustainability, and our food system has the role to play. It's not, of course, the only one, but a significant one. And in this light, one thing we can do is move towards more plant-based foods, using more plant-based proteins, and that in mind, the main aim in our work package has been to assess the impact of a sustainable plant-sourced diet, or on the other hand, a fermented side stream originated food on human gut microbiota or microbiome, and then health, if that has any health consequences. So we've had 3 major objectives, and 2 of those has consisted of a randomized clinical trial, which is like the ultimate stress test to see if any of the great things Minna presented to you has really effect on humans, and that's and it's actually quite a hard task to really show in a controlled conditions that something changes, because we humans are individuals with different characteristics, and there's really quite a big variation in consequences of diet between different persons. And then we had an objective 3, which consisted of cell culture studies looking at the underlying mechanisms. And I'm not going into details, just showing like the very major results we have. Just for your background info, you need to know what sort of trials we have been carrying out. So the first task involves Helsinki trial, which is a randomized control trial where we replaced part of animal-sourced proteins with plant-sourced proteins in healthy working-age adults. And this was quite a long intervention, 3 months. We had 3 dietary groups. The first one, which I call animal, representing a current average Finnish diet, where 70% of proteins come from animal sources and 30% from plant sources, mainly whole grains. And then we put all the animal-sourced proteins together and started slicing and replacing with plant-based sources, which consisted of faba bean, peas, so legumes in general, some soy, nuts and seeds, and that made sort of up the animal replacement of animal protein. Then we had 50-50 group where there are equal amounts of animal and plant proteins, and then we had plant dominant group with 30% of animal-sourced proteins and 70% of plant-sourced proteins. So just for your notice, none of these diets were vegan diet, because we didn't want to be sort of a bordered calculating amino acid intakes. And so we supposed that all of these diets were sort of safe diets in terms of nutrient intakes. So we collected quite a lot of samples and data. Even when we gave most of the foods, so representing 80% of energy consumed by the study, we still wanted to see what else they ate. So they were filling food 4 days, food diaries. So we know exactly what they ate before they started the intervention and during the intervention. What we didn't control were vegetables with no protein content, fats, so fat spreads and oils, sweets, and alcohol intake. We took blood samples, we took urine and stool samples, and then we also carried out an oral glucose tolerance test to subgroup of the volunteers. So basically we used a whole diet approach, meaning that we controlled the protein intake, the concentration of protein in every diet. The aim was 17 energy%. But that means that still different protein sources come with different other nutrient components. So it means that that really nutrient intakes changed. And we could see, for example, an increase in fiber intake. And we were expecting, because this is actually quite a drastic change in a diet, so we were expecting quite a lot of results on gut microbiome, for example. But basically, we couldn't see any change in gut microbiome diversity. And the changes that happened were mostly in the plant group, and they were at genus or general level, and partly depending on the characteristic of the person's gut microbiota or metabolic properties of gut microbiota, which I can't go into more details because of the lack of time. But it seemed to be quite complicated. It would take like a half an hour to explain what we saw, and so I won't go to that. We then measured quite many gut stool metabolites in order to connect gut microbiome to metabolic health of these volunteers. So we measured, for example, fecal N-nitroso compounds, a risk factor for colorectal cancer, and because they are supposed to be genotoxic, we measured genotoxicity. We could see actually a quite drastic decrease in N-nitroso compound concentrations in the stool samples, but we couldn't see any major effect on genotoxicity. We could see a reduced concentrations of secondary bile acids, increase in primary bile acids, and this is considered good, at least from the prevention of colorectal cancer point of view, because secondary bile acids are thought to be detrimental to gut cell walls. We also measured some polyphenol metabolites, because plant-based food is known to contain loads of different polyphenols, and they are supposed to have health effects. We did see some changes in the profile of polyphenol compounds or metabolites. We are not quite sure whether we can directly connect them to any health effects. We did measure blood lipid profile, and that was improved in terms of LDL cholesterol that was decreased in the plant-based groups, and there was also a small but significant improvement of insulin sensitivity with the most plant protein group. So, yeah, something we expected and something we didn't expect, and I'll come to the interpretation of the results later on. Then in task 2. We had another trial which was with a different principle, so that there actually that was like not a whole diet approach, but like a supplement of rapeseed seaweed product, And here, once again, this was done here in the University of Copenhagen. This is once again a double-blinded RCT with 6 weeks intervention, and the treatment was 5 grams of fermented canola seaweed preparation or a placebo. And in a similar way, fecal samples, blood samples were collected at the baseline and during the intervention, and then analyzed. And yes, so there were quite high hopes to see beneficial effects, but really it seems not to be so. So basically there were no changes in gut microbiome composition. There was no changes in the oral glucose tolerance test, which was one of the major outcomes. There were no changes in other clinical parameters, for example, a marker of inflammation. There was no change in blood lipids. And then briefly about the in vitro studies, which were actually quite interesting. So some polyphenol metabolites and bile acids recognized in the Helsinki trial were further studied with these cell lines, and this was done in NMBU in Oslo. And here, basically, what was observed is that some polyphenol metabolites had a weak but consistent dose-dependent effect on inducing T-cell proliferation, which can interpret it as a positive effect. So perhaps inducing like a positive immune response in the gut. Also, there were the same polyphenol metabolites were associated with antigen presentation in macrophage differentiation assays, and this is also could be interpreted as positive results. On the other hand, we could not see any changes in aryl hydrocarbon or bile acid receptor signaling, or nor in the measure of epithelial barrier function. But yeah, I think these results were quite interesting. So what have we learned about trying to change adult gut microbiome by dietary means? First of all, it's not easy. It's actually quite, of course, it's what we aim at. So if we think that we can change microbiome at the level of diversity, then I would say that that won't happen easily. It's quite, so adult microbiome sort of overall composition seems to be quite stable, and not even a drastic dietary change as our Helsinki trial could change that. There are most certainly genus level alterations which could be interesting, particularly if we relate them to functionality of these bacteria. Yes, here really the dose is important. If the dose is too low, then there's no effect. And at least Dennis, who was responsible for the Copenhagen trial, thought that really the dose wasn't enough. But we could see quite many significant changes in fecal metabolites, either in the concentration or in the profile, and once again, if dose is high enough. And in terms of health, I think the metabolite profiles are actually more important than the gut microbiota diversity, because it's the metabolites that mediate the health effects. So I think in the future, I think we should really concentrate more on the microbiome metabolic activity, and because I think that's what matters. And clearly there seems to be differences in the metabolic activity between individuals. But that's also quite clearly needing quite a lot of new research. of course, have we contributed to SIMBA's overarching objectives in terms of exploitability? I think it's really, really important to increase the knowledge and understanding of the role of diet's impact on gut microbiome and health, because, I mean, I think it's still early days. You've seen how many new publications about gut microbiome there is coming out every day, but still, I think we know next to nothing. And for a scientist, that might be a happy situation. There's still not a lot to explore, but of course, we'd like to work so that we could use diet to optimize gut microbiome activity and promote health. And really, I think what we can say is that many of the chronic disease markers reacted. Let's say blood lipids, insulin sensitivity, and I think that's good, but we still sort of need to study what's the role of gut microbiome there. Yes, so about the exploitability, I think that's perhaps the scientist is not the best person to say how exploitable the results are. And I think we should be able to target gut metabolic activity and prevention of chronic diseases. I think that's the aim. But to what extent we can do it today is, I'm eager to hear what the panelists think about this. I think some of the cell culture results implied that there's a possibility to impact gut barrier function and immune responses, which consequently have a health effect. And then what I think is very important to consider is what are the doses and what kind of products are needed to have an impact and to be exploitable, let's say, in food development. Yeah, and I think that's it. Thank you.
Thank you, Anna-Maria. And I think that we have to continue to the next presentation now. Okay, one question, yes.
Thank you very much. It's a shame you didn't have the extra half an hour to tell more about the microbiome. I wonder whether you have looked or thought about whether you had like strain replacement you wouldn't see in the microbiome. So there was a change, actual change in the microbiome, but the level of resolution was not enough, the 16S, or whether you measure activity and the activity change in different groups. So I don't know.
Yeah. So actually, we did. We analyzed the gut microbiome both using 16S and metagenomics, and basically the results agreed with both of these techniques, and the functionality things we've extracted from the metagenomics data.
But you didn't do like meta-transcriptomics to see which groups were active at different points of the treatment, or you think it's a good idea?
Yeah, well, yes, we did, but so we basically have the baseline values or the results and the what happened at the intervention. So yes, so I'm not quite sure what are you aiming at? So
no, well, actually, I would need more time also, but I think that for me, and I guess for you too, it was a surprise to see that there was no changes. And I was wondering whether the resolution, because you said that below the general level, the genus, and this is a very high, below this level you have species and strains.
Yeah, sure.
And maybe this you couldn't see even with metagenomics. I was wondering because I find it amazing that we can talk later. Thank you very much.
Let's change words afterwards. Thank you.